morning, learners. I am Sanda. I'm back with you again, looking at the topic of genetics and inheritance. Today, we're going to be focusing on dihybrid crosses. And let us look at the examination guidelines to see what is it that we are supposed to cover under this topic. Under dihybrid crosses, as you can see there, it says we must look at Mendel's principle of independent assortment, which is basically now another law of Mendel. If you remember, we talked about the principle of segregation as well as the law of dominance. Then we must also look at the dihybrid genetic problems like we did with the monohybrid crosses. We did some problems involving monohybrid crosses where we were crossing one characteristic. So we are going to be looking at dihybrid genetic problems. Then lastly, we must also be able to determine the proportion or the ratio of genotypes and phenotypes when we are doing these genetic problems, as we did with the monohybrid crosses. Now, what is a monohybrid cross? Because we've been talking about, what is a dihybrid cross? Because we've been talking about monohybrid crosses all along. Now, a dihybrid cross, if you think about di, what does di mean? Di means that you are looking at two things. So if it is a dihybrid cross, you are basically crossing two individuals where you are concerned with the inheritance of two characteristics simultaneously. If you remember, with monohybrid cross, we were only looking at the inheritance of one characteristic. So here we are looking at the inheritance of two characteristics. So let us use example of the pea plants that Mendel used in his experiments. If you remember, when he was doing the monohybrid crosses, he looked at 7 characteristics, and one of those characteristics was the shape of the seeds as well as the color of the seeds. So we're just going to use the example of seed shape and seed color as we looked at before monohybrid crosses. Now we will be looking at the passing of these two characteristics at the same time. So if we look at these two characteristics, in seed shape, there were two variants of the seed shape where the seeds could be round or they could be wrinkled. And then we also saw that in seed shape, the round seed shape was dominant. Hence, we are using a capital letter R to represent round seed shape over the wrinkled seeds, which were recessive, and therefore for wrinkled seeds we are using the small letter R. Then with the seed color, there was yellow seeds as well as green seeds, where the yellow seeds were dominant over the green seeds. So for yellow seeds, we're going to use the capital Y, and for the green seeds, we are going to use the small letter Y. Now the question says, what are the expected phenotypes in the F2 generation when a pure breeding plant with round and yellow seeds is crossed with one that has wrinkled green seeds? So if you look at this table lays out these characteristics. So we are looking at two characteristics. One is seed shape and the second one is seed color. So it is a dihybrid cross. Now with each characteristics, there are two variants of that characteristic. We refer to them as traits. So for seed shape, you can have round seeds and you can have wrinkled seeds. So round, because it is dominant, we are going to use the capital letter R, or the allele representing the round seed will be represented by the letter R, capital R. And then for wrinkled seeds, because it is recessive, we are using a small letter R for wrinkled seeds. Then for seed color, two traits there. They can be yellow or they can be green. Yellow is dominant, and therefore we use a capital Y for yellow. And then green is recessive, and we use a small letter Y or the green seeds. So let us look at a cross which involves the passing of those two characteristics. Now, as we did in the monohybrid crosses, Mendel started with pure breeding plants, so meaning that the plants were homozygous at the beginning or in the first generation. So here we are crossing round and yellow seeds with wrinkled and green seeds. Round and yellow are the dominant characteristics. So for the genotype of the round and yellow seeds, we use capital R for the round characteristic and capital Y for the round for the yellow seeds. So you will see here, just like in monohybrid crosses, for each characteristic that we are looking at, there are two letters. Remember, you get one allele from each of your two parents. So for round seeds, we are using two capital R's, and for the yellow seeds, we are using two capital Y. Same thing with the wrinkled and green seeds. It's two small r's, two small y's. Then the next thing that happens is that now the alleles in those in the parents, they segregate during meiosis for the formation of gametes. So you will see now we have four letters that we must work with. So we must look at all the possible combination of those four letters and how they can lead to the gametes here. So you will see I have used colors so that you can be able to see what matches with what. So remember, we are looking at two characteristics. So each gamete that is produced, it must have alleles for the two characteristics. So now let's look here. In this genotype of capital R, capital Y, there are four combinations that we can get here. The first R of the genotype can combine with the first Y to form one gamete, which is going to be capital R, capital Y. Then the first R can combine with the second Y if you follow the purple lines, which also gives us capital R, capital Y. Then the second R can combine with the first Y to give us again capital R, capital Y. Then lastly, the second R can combine with the second Y, which gives us capital R, capital Y. So those are the gametes of the first parent. But here, note that the gametes are all the same. And we are going to talk about what we can do if the gametes are all looking the same. In the same way, for the second parent with the wrinkled and green seeds, we work out the gametes like we did with the first parent. So the first small r can combine with the first small y so that we get a small r and a small y as a gamete. And then the first small r can combine with the second small y, then we get our second gamete. And then the second r, small r, can combine with the first small y to give us again another small r, small y in the gamete. And finally, the small r, the second small r, can combine with the second small y to give us the small r and small y in the gamete. All of these gametes are the same. So normally you would then use a Punnett square, because I would advise that for the dihybrid cross, you use a Punnett square. It's much easier to work out the gametes if you are using the Punnett square. But because here the gametes, all the gametes from the one parent are the same, and also from that parent are the same, so we just look at one gamete here and the other gamete from that second parent and cross those two only. We don't have to do the whole Punnett square here. So if we cross the capital R, capital Y with the small r, small y, then the genotype of the offspring is going to come out as capital R, small r, and capital Y, small y. Please note that I am putting the same letters, or the letters that are coding for the same characteristic, together. So the R's, they code for the seed shape, and the Y's code for the seed color. And also note that we always start with, when we are writing the genotype, we always start with the capital letter or the dominant allele. So it's capital R, small r. It's not small r, capital R. So it's capital R, small r, capital Y, small y. So when we translate that into the phenotype or the physical appearance of these plants, it becomes all the plants that come out of this cross, they are all round and yellow, meaning that we have now the dominant characteristic is masking the effect of the recessive characteristic. So the small r is masked by the big R, so that the seeds are all round, and the small y is masked by the big Y, so that all the seeds are yellow. So let's carry on. And the next slide is just to highlight just use it in the form of a Punnett square. It's this very same cross, but you can see now when when you do a Punnett square, it is much more neater. Same here, we're starting with round and yellow parents crossed with wrinkled and green parents. Genotypes, these plants are homozygous, or they are pure breeding. So you have capital R, capital R, capital Y, and then you have small r, small y, small y. Then meiosis occurs, which brings the principle of segregation, and therefore your gametes will be capital R, capital Y from the first parent. You don't have to write all of them if they are all the same. In the same way, for the wrinkled and green parents, the possible genotype is small r and small y. So that is the only possibility there. So when we fertilize these two, or they fuse together, then the genotype of the first generation offspring is going to be capital R, capital Y, small y, which gives us the phenotype round and yellow seeds. So now the question was, what is the expected phenotypes in the second generation? So now let us look at how what are we crossing in the second generation. Now, like we did with the monohybrid crosses, we took, or Mendel took, the offspring of the first generation and made them the parents of the second generation by allowing them or by crossing them, or by allowing them to self-fertilize. So now you will see in the P2, we're starting with round and yellow parents, which came from the first generation. The genotypes of these two parents is the same. They are heterozygous for the characteristics we are looking at, meaning that the genotype is capital R, small r, capital Y, small y. So if we look at the gametes, now what are the possibilities with the gametes? We are just going to look at the top part here, because the two parents are the same, so the gametes are going to be the same. So if we look here, remember we said for each gamete, it must have the allele for each of the characteristics that we are investigating. So it must have an R, it must have a Y. So how do the alleles combine? The first possibility is the capital R can combine with the capital Y, and it gives us that gamete over there, which is capital R and capital Y. Then the second option is the capital R can combine with the small y, and then it gives us a gamete that has capital R and small y. The other option, the third option, is the small r can combine with the big Y, and therefore you get that gamete over there, which is small r and big Y. Then lastly, the small r and the small y can come together and they give us a gamete that is small r, small y. So for the second parent, here are the gametes on the left-hand side here, and they are exactly the same as the ones that we put up here for the first parent. Now, the next thing for us to do is to look at the fertilization, which happens now inside the Punnett square. So when we are doing the fertilization in a Punnett square, we look at these gametes at the top here. We start first with the first doing or filling in the first column. This column would have been empty. We fill in the first column, so going from the top to the bottom. So the capital R, capital Y will fuse with this capital R, capital Y, and then it gives us capital R, capital R, capital Y, capital Y. So second row, we are crossing capital R, capital Y with capital R, small y. So therefore the offspring becomes capital R, capital R, capital Y, small y. Then in the third row, we are crossing capital R, capital Y with small r, capital Y, and therefore our offspring becomes capital R, capital R, capital Y, capital Y. And then lastly, we have capital R, capital Y with small r, small y. So we are done with the first column. So what do we do next? We go to the second column, to the gamete in the second column at the top. We go row by row to fill in the genotypes of the offspring, which you can see in that whole Punnett square. So I'm not going to go through all of them. So when you are doing it on your own, you go first column and then row by row, second column and then row by row until you get to the last column, doing exactly the same thing. So what we can notice here is that each dihybrid plant, when we are saying dihybrid plant, it means that the plant is heterozygous for both of these characteristics that are being investigated. So each dihybrid plant, for example, capital R, small r, capital Y, small y, produces 4 gamete types of equal frequency. So you've got one of each of these different gametes here. So they are produced equally. So let us carry on now with the dihybrid process to interpret now what is inside the Punnett square. To make it easy for you, I have labeled these genotypes with numbers so that you can see the ones that produce the same phenotype. So in this Punnett square here, this is only the inside of the Punnett square where the offspring are. You will see that I've got a couple of genotypes which I have labeled number 1. So if we look at those that have got number 1, let's look at the first one. It's capital R, capital R, capital Y, capital Y. So this translates to the phenotype which is round and yellow. Then the second number 1 is capital R, capital Y, small y, which also translates to round and yellow. So you can count how many of these offspring have got capital R in their genotype, and they've got at least one capital Y in their genotype. So there is 1, 2, 3, 4, 5, 6, 7, 8, 9. So in this cross, we end up having plants that 9 of these pea plants will have round and yellow seeds. So now let's go to the ones that are labeled number 2 and interpret what does the number 2 translates to. So you can see in this one, how many number 2s do we have? We've got 1, we've got 2, and then we've got 3. Now let's look at what does that represent. It's capital R, small y, small y. So meaning that here we've got round seeds which are wrinkled. The second one is capital R, small r, which represents round, and then the other, the y's, you've got 2 small y's, which also translates to round and wrinkled. So we can say in this Punnett square, we had 3 seeds that were round and green. Then let's go to number 3. How many number 3s do we have? You can see we've got 1, 2, 3 number 3s. And they all translate to the same phenotype, which is going to be wrinkled and yellow. It's also wrinkled and yellow over there. And over here, it is also wrinkled and yellow. So we have got 3 offspring that have got 3 that have got wrinkled and yellow seeds. And then we've got number 4. You see that there's only one number 4, meaning that there's only one individual that has got that genotype, and therefore there's only one individual that has got the genotype, the phenotype wrinkled and green seeds. So number 4 represents wrinkled and green seeds. Now I want you to remember, what did we cross at the beginning? At the beginning in P1, we crossed 2 parents, one with round and yellow seeds, and the other one had wrinkled and green seeds. So what do we see now from the results of this cross is that we are now having our offspring in the ratio of 9:3:3:1. So that is the phenotypic ratio. 9:3:3:1. Another thing that we are noticing here is that there are 2 new combinations that were not there in the parental generation. That is the round and green seeds, as well as the wrinkled and yellow seeds were not there at the beginning, meaning that we can see that the alleles for round and green seeds, they did not stick all the time with with all the alleles in that genotype of the parents. So they sort of assorted independently. We will discuss the law or the principle of independent assortment at a later stage. So again, just for you to have a visual picture of these seeds, we are talking about round seeds that are yellow, as well as round and green seeds and wrinkled and yellow seeds. So if you look here, it's exactly the same thing that you can see over there. Now I used triangles for you to be able to see clearly these different phenotypes in that Punnett square. So this big triangle, red triangle, it encircles all the ones that have got round and yellow seeds. If we count how many, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. So the big triangle is mainly to highlight the round and yellow seeds. Then we have this brown one here. The brown one is mainly to encircle the ones that have got green and round seeds, or round and green seeds. So how many round and green seeds we see here? It's 1, 2, 3. Then we have one that encircles the wrinkled and yellow seeds. How many wrinkled and yellow seeds? We have got 3. And then finally, the black one at the bottom, it encircles that one that has got wrinkled and green seeds, which is only one of that. So that is how the ratio of the phenotypes come in a dihybrid cross. And also note that there are 16 possible offspring as opposed to 4 of possible offspring in a monohybrid cross. So just in summary, the characteristics, so that we can also find a way of summarizing the genotypes and the phenotypes that we get in the dihybrid cross. We said that R is dominant. So which is round shape is dominant, so we use a capital R. And then for wrinkled seeds, we use a small r because wrinkled is recessive. And then also for the color of the seeds, yellow is dominant, so we use a capital Y, and green is recessive, we use a small letter y. So we can actually group the genotypes. So all genotypes that have in them a capital R and a capital Y, even if there is a small r somewhere. If it's got capital R and capital Y as one of the two letters there, then the offspring will have round and yellow seeds. Then secondly, if we have capital R somewhere in the genotype and we have got two small y's, it means that the seeds are going to come out round and green. If we've got two small r's and a y somewhere in the genotype, we are going to get wrinkled and yellow seeds. And then if we've got two small r's, two small y's, then your seeds come out wrinkled and green. Now let us talk about the Mendel's principle of independent assortment, which I said it is an addition to already two other principles or laws that we talked about earlier in the monohybrid crosses, which is the principle of segregation as well as the law of dominance. Now, your dihybrid crosses basically reveal this principle of independent assortment. We are going to see how does that happen. This principle, it states that the alleles of a gene segregate independently of the alleles of the gene for another characteristic. Now remember here, we are looking at a dihybrid cross. So we've got two characteristics that are coded for by two different genes. And for each gene, we have got two alleles. So the alleles for the gene, for example, for seed color, will segregate independently of the alleles of the gene for seed shape. And then when the alleles come together during fertilization, it says that the alleles for each gene therefore come together randomly during the formation of gametes. We are going to see that if we are looking at this example here again, where we are crossing the round and yellow seeds with the wrinkled and green seeds. So here, like, let's follow what Mendel did when he was doing this cross. In the first generation, or in P1, which is represented over there, he crossed pure breeding plants, one with round and yellow seeds and the other one with wrinkled and green seeds, as you can see in the picture. Then in the first generation, or in the F1 generation, all the offspring that were produced were having the genotype capital R, small r, capital Y, small y, which means that they were all round and yellow. Then the next thing that was done is it was to self-fertilize the offspring of the first generation, which was represented by the capital R, small y, capital R, small r, and capital Y, small y, which is shown over here as we did in the other cross. So there's the one parent and there's the other parent. These are the gametes of the first parent over here. What you can see here is that we said there are four gametes that are produced in a dihybrid cross, and they are produced with equal frequency. So there is a 1/4 chance of getting this gamete that has got capital R, capital Y. There is a 1/4 chance that has that produces the gamete with capital R, small y, and also same thing, small r, capital Y is also produced in the same frequency. So they are equally produced, these gametes, for both of these parents. So there's your results in a Punnett square, exactly the same as what we did in the previous slide. So what we are trying to show you here is that when the alleles of the genes segregate, they are segregating independently of each other. Hence, we can see that the round and yellow and the wrinkled and green combinations observed in the parents, they did not stay together in the offspring, meaning that the alleles, they segregated independently of each other. Hence, we had that option of getting the wrinkled the round and green seeds, as well as the wrinkled and yellow seeds, which were not there in the parents. So that is what the principle of independent assortment is showing us. Again, here I've used this picture just to show you this principle of independent assortment, where it says here the different genes are sought, or they are passed into the gametes independently, because they are located on different chromosomes, which align randomly at the metaphase plate or at the equatorial plane during meiosis 1 or during metaphase 1. So if you look here, starting at the top here, this would be like a representation of the nucleus of a cell, with the chromosomes that are carrying the alleles, capital R, which is carried in that red chromosome, and the other capital Y in that smaller, shorter red chromosome. And then you have your small r in that blue chromosome, which is longer, and then you get your small y in that blue chromosome, which is shorter. Now, during metaphase 1, during metaphase 1, when these homologous chromosomes align at the equator, you must note now the way or the different arrangements in which they can arrange themselves. Just think of the fact that these two parents have the same genotype. It's capital R, small r, capital Y, small y. Same thing on this side. But look at the arrangement of the chromosomes, of the homologous chromosomes at the equator during metaphase 1. In the first cell, what we can see is that all the chromosomes, all the blue chromosomes, have arranged on the one side of the equator, whereas in the second cell, they have been mixed up. So there you've got the blue and the red one, and then here you've got the red on this side and the blue on the other side. So that is what we are saying with the random arrangement of chromosomes at the equator during metaphase 1. And we have mentioned before that this random arrangement is what brings about variation in the gametes that are produced during meiosis, which also then leads to variation in the offspring. So as they have randomly arranged, now obviously the next step will be the anaphase 1, where they move to the opposite poles. So now they've already been arranged in that manner. So when this cell splits into two, it's the blue chromosomes are going to go into the one gamete with the alleles that they are carrying, in this case the small r and the small y. Then the red ones are going to go into the other gamete, that is the capital R and the capital Y. So this is now the assorting part. That is the assorting part. And then, so which means that the order in which they were arranged is not going to change when they now move from the equator to the opposite poles during anaphase 1. And then, so that is how they will look like at the beginning of metaphase 2. So here, this is how they were arranged, and this is how they look like in the gamete. So the way they were arranged at metaphase 1, they stay like that until they are the gametes are produced. So all the blue ones with the small r and the small y will form the two gametes there. Same thing here. So what we are saying is random arrangement that results in variation, or that produces variation. The assortment, when they move to the opposite poles during anaphase 1 or during anaphase 2, it doesn't result in variation. So please note that when you are asked that question about the sources of variation, it is not random assortment. It's not, we say it is the random arrangement of the chromosomes at the equator. So this diagram here, it is just to illustrate to you the principle of segregation, which happens in a dihybrid cross as well. So there's your parent with the genotype capital R, small r, capital Y, and small y. Inside the cell are the chromosomes that are carrying the different alleles of that parent. Now here it shows you the arrangement of these chromosomes at the equator, and they segregate each of these chromosomes. They segregate into the different gametes that will be produced. So this is showing you at metaphase 2, this is how the cell would be looking like, with the chromosomes arranged at the equator, and then the segregation happens so that the one gamete will have the small r, small y, and here they will have the capital R, small y. So from this parent, these are the possible combinations in the gametes that we can get, which happens as a result of segregation. Same thing here, it's showing you all these 3 laws of Mendel. In the beginning here, it's showing you a different example where 2 heterozygous pea plants were crossed, and both had tall and purple flowers. As you can see in the picture, the plant is tall, it has got purple flowers, and on this side it's a tall plant with purple flowers. So with segregation, we are saying that the alleles are segregated, or they separate during the formation of gametes, so that each gamete will have one copy of each gene. So you can see here, this side, or if the cell split in half here, that gamete has got those 2 chromosomes. And then that second gamete has got those 2 chromosomes. So that is the segregation of chromosomes, and they independently assort, meaning that they move independent of each other into the different gametes. So when fertilization occurs, these gametes can combine randomly to form the zygotes, or to form zygotes with 2 copies of each gene. So in this picture here, you can see we have got a tall plant with purple flowers because of the combination of the alleles. And then the second one has got a tall plant with white flowers. And the third one gets a short plant with purple flowers. And then the last one is showing you the short plant with white flowers. So you can see segregation as well as independent assortment, and the principle of dominance is displayed in this picture. So those are 3 important laws of Mendel. Now let us do an activity. It's a 3-part, or it's got 3 questions. Now in this activity, it says in rabbits, gray hair is dominant to white hair. So we're looking at one characteristic first. Here is the color of the hair. And then it says in rabbits, the black eyes are dominant to red eyes. So the second characteristic we are investigating here is the eye color. So again, putting all this information in the table, the 2 characteristics is hair color and eye color. And the traits or the variants of these characters is, for hair color, we've got gray hair, and we have been told that gray hair is is dominant to the gray hair is dominant to the white hair. So gray hair, in terms of the genotype, there are two possible genotypes there. It's capital G, as well as capital G and small g, meaning that if the rabbit has got gray hair, they can be homozygous or they can be heterozygous. Heterozygous meaning that there are two different alleles forming the genotype of that individual. With white hair is recessive, so there's only one option there, which is that one of having two small g's. With eye color, we can have black eyes and red eyes. Black eyes is dominant, so it will have a capital B when the individual is homozygous, or it will have a capital B, small b, when the individual is heterozygous. Then for red eyes is recessive, so it is represented by two small b's. Now let's look at the questions linked to this information. The first question, it says, what are the phenotypes or the descriptions? How does the organism appear like of the rabbits that have got the following genotypes? The first genotype is capital G, small g. Remember, we are looking at hair color, which is either gray or white. So the small b's represents recessive color of the eyes, which is red eyes. So the first individual is going to be having gray hair and red eyes. The second one has got two small g's, meaning that they will have white hair, and two small b's, which means that they will have red eyes. So the next phenotype is white hair and red eyes. Then the third one is two small g's, meaning that they will have white hair, and two capital B's, meaning they will have black hair. So it's white hair and black eyes, not black hair. It's black eyes. Then the last one is capital G, small g, capital B, small b, which gives us gray hair and black eyes. Then number 2 says, a male rabbit with the genotype capital G, small b, is crossed with a female rabbit with the genotype capital G, small b, capital G, capital B, small b. And then this Punnett square is set up out below, as you can see there. And it says you must fill it out and determine the phenotypes as well as the proportions in the offspring. Remember, in the exam guidelines, it says we must look at the ratios or the proportions of the genotypes and the proportions of the phenotypes. So this question is asking you to do exactly that. So if we look at filling in the Punnett square, we do it exactly like we did before. It's going first column and then matching with the gametes on the left hand side, row by row. So for the first one, it's going to be capital G, capital B, small b, and so on. So that is how they appear. Then we go to the second column and match it with the gametes on the left hand side. So this is what we get there. So you will be doing this on your own. And in the third column, we get those offspring. We continue with that until we have filled in the whole Punnett square. So that is the genotypes of the offspring. Note that there's 16 squares inside, so there's 14 possible offspring there. They may have the same phenotype, or they may even have the same genotype. As you can see here, in the first two rows, the genotype of the offspring is the same, as well as in the last two rows, the genotype of the offspring is the same. So meaning that the first two rows, the phenotype will be the same. So let's look at what is the phenotype there. We've got capital G, small g, capital B, small b, which translates to individuals that are gray and black, and so on. And then we have for this one here, it's capital G, small g, and then small b, which means that the individuals will be gray and red. So we carry on like that, filling in all the different phenotypes. I don't know why is the other ones not showing here. So it is asking us how many out of the 16 have gray fur and black eyes. So all these ones in the first two rows, they are gray and black. So meaning that there is 8 individuals that are gray and black. Then it says how many out of the 16 have gray fur and red eyes. As you can see, the last two rows is made up of the gray and red. So meaning that we have got 8. How many out of the 16 have white fur and black eyes? Do we have any? There is none. How many have got white and red eyes? Then it gives us 0 again there. So though you can see now it starts to fill up the Punnett square. So all the offspring in the first two rows, they are having gray fur and black eyes. So hence we are having 8 over there. Now let's go to the next question. Now the next question, I keep moving myself here. The next question says, a male rabbit with a genotype capital G, small g, capital B, small b, is crossed with a female rabbit with the genotype capital G, small g, capital B, small b. There is your Punnett square, and it says fill it out to determine the phenotypes and the proportions of the offspring. So in the same way we fill out this Punnett square with the offspring's genotypes, and that is how it goes. Column by column, first column, and then all the gametes in the rows, and you go to the second column, and you go to the third column, and finally the fourth column. So here's your genotypes of the offspring from that Punnett square. And now let's look at the phenotypes. So the first one there, that it should be that one, but it looks like my animations, I did it in the wrong order. But that one, if you look at it, it's capital G, small b, small b. So that's gray and red. That is also gray and red. Gray and red. Sorry. This one is going to be white and black, because we have two small g's, which represents the recessive allele or the recessive characteristic, which is white fur, and then the capital B represents the dominant characteristics, which is black. So it's gray, it's white hair and black eyes. Same thing, that one will also be white and black. That will also be white and black. And this one here at the bottom is the one that is going to be white and red. So how many have got gray fur and black eyes? All the ones that at this moment are not labeled. The labeling will come later. You can count 1, 2, 3, 4, 5, 6, 7, 8, 9. Those will have black and gray. You will have gray fur and black eyes. 16. How many have got gray fur and red eyes? That will be 1, 2, 3, as you can see in the Punnett square. How many out of 16 have white fur and black eyes? As you can see, we've got 1, 2, 3. How many have white fur and red eyes? There is only 1. Sorry. Then let's look there at the top again to just find the 9 that have got gray hair and black eyes. There is your gray and black eye. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's your complete Punnett square. Just a second example taken from a past exam paper. Here it says the leaf color in a plant is controlled by 2 alleles. There's the allele for the green color, which is dominant because we see that it is represented with a capital letter, and yellow leaf color is recessive because it is represented with a small letter G. That is the first characteristic being investigated here, is the leaf color. Then it says thorns on plant stems are controlled by two alleles, which represent one. The presence of thorns is represented with a capital letter T, meaning that having thorns is dominant to having no thorns. So no thorns, as you can see here, it is represented by a small letter T. So first characteristic is leaf color, and then the second characteristic is the thorns on the plant stems. Now it says two plants with the genotypes capital G, and small t, capital G, and capital T, was crossed with small g, and capital T. And then it says their offspring were then left to pollinate each other, meaning that we started with two pure breeding individuals here. And then we took their offspring, meaning that the offsprings from here will have heterozygous genotypes. That means that the offspring that are going to be crossed and show the results in this Punnett square would have been capital G, small g, and capital T, small t, which you can see in the representation of the gametes there. You have capital G, and then you also have a small g there. You have a capital T, and then you have a small t over there, meaning that this parent here was heterozygous for this leaf color and for the presence or the absence of the thorns. Now it says the table below shows the possible genotypes of the offspring of the second generation. Genotypes I, or number 1, and number 2 have been left out. So there is number 1, which doesn't have a genotype there, and there is number 2, which also doesn't have a genotype. Now let us look at the questions. Question 2.1, let's put the table or the Punnett square there again so that we don't keep going back to the previous slide. Give the genotype of I or 1. There is 1 where it's located. So now if you look at the gamete at the top here, it's got a small letter G and a capital letter T. And then the second one that matches it, this is in the second row, so the gamete will be a capital G and a small t. Now when we combine these two, it's going to be the capital G and the small g from there, and then the capital T and the small t from there, so that the genotype of I is capital G, small g, capital T, small t. Then B says we must look at the phenotype of 2. Now this is 2 here. Firstly, before we can determine the phenotype, we have to know what was the genotype of that offspring. So that genotype would have been small g, because we are combining this one and that one. So it would have been small g, which is recessive for the leaf color, as well as small t. So this parent or this offspring here is going to have yellow leaves. Remember, yellow is recessive to the green leaves. And then no thorns is recessive to the presence of thorns. So this plant has yellow leaves and it doesn't have thorns. Then in 2.1.2, it says list the 4 genotypes of the offspring of the second generation that would be phenotypically different from the original pair. The original pair was green with thorns and yellow without thorns. So we want genotypes that represent offspring that doesn't have green with thorns or yellow without thorns. So the possible genotypes would be these ones. Here, that means that if we are saying capital G means that it is green, and then small t means that it is without thorns, whereas at the beginning we crossed green with thorns. So it's capital G, small t, or we can have capital G, small g with two small t's, which is also green without thorns. Then the other option is to have yellow with thorns. Remember at the beginning we crossed yellow, sorry, we crossed yellow, no, we crossed green with thorns and yellow without thorns. So here we are having yellow with thorns, which was not in the parental generation. Or it can be two small g's, capital T, and a small t. So that is basically the end of our lesson. We always have a worksheet that we must do that is linked to this activity. So you will get that worksheet on dihybrid crosses. And the lesson planned was to have mutations as well, but because this is such a long presentation, so mutations will be done in the lesson for tomorrow. So you will do the worksheet that goes with the dihybrid crosses, just to consolidate what you have learned here. And remember, I said that there are two websites that have got nice resources for you in terms of the exam papers from the previous years. You can go to www.ecexams. All the letters are small letters, and there are no spaces in between. So it's www.ecexams.co.za, or you can go to www.ecurriculum.co.za. Thank you for listening. I will see you tomorrow.